Okay. Hello, everyone, and welcome to our seminar series. So one of the major objectives of genetic improvement is to obtain resistant genotypes. To this end, plant breeders have traditionally exploited resistant sources taken from existing resistant varieties or from wild sources or wild relatives, which are usually conserved in gene banks such as the International Cocoa Gene Bank. The work of the pathology team at the Cocoa Research Center centers on the evaluation, characterization, and exploitation of this genetic diversity for resistance to pathogens in wild or primary germplasm, which is held at the ICGT. This presentation, therefore, will highlight some of the accomplishments made towards two important diseases of cocoa, which are black pod disease caused by Phytophthora parmivora and witch's broom disease caused by Monilioptera perniciosa. Here I'm just giving you a brief overview of the steps which have been taken over many years of work that have led towards developing, have been put towards developing varieties to support cocoa farmers. So the two major diseases, black pod and witch's broom, contribute to significant losses as shown in this table. Together, they account for $1.5 billion in losses annually. And also noteworthy to us in the Caribbean and in Latin America is that these two diseases are two of the top 3 diseases of cocoa worldwide. While black pod is ubiquitous around the world, witch's broom and frosty pod disease, although confined to the Western Hemisphere or the Americas, as we say, are rapidly spreading to new territories within the Caribbean. Globally, we can see that cocoa diseases have wreaked havoc on many cocoa sectors worldwide. We can see that this impact, if you ask cocoa farmers what to name as their major constraint to productivity, I'm pretty sure the answer is always diseases. And it is estimated that diseases account for over 40% of losses in production on average, and as much as 90% for some diseases. So what are the strategies that are usually employed to control these diseases? Traditionally, we've used cultural practices, which includes field sanitation, shade management, drainage issues, chemical and biological control. But it has always been recognized that the genetic improvement or the genetic approach, although long-term, is the most cost-effective and sustainable of the strategies that we could employ in our quest for disease control in cocoa. This slide looks a little busy, but it shows the pipeline that we have used in our journey towards developing new varieties for our cocoa farmers. And our beginning starts with, of course, the gene bank. We start at our gene bank, which is the source of our cocoa genetic diversity, which is imperative or central to the deployment of resistance in farmers' fields. And in order to exploit that resistance, we must identify these sources of resistance. And here, we must do development of optimized and validated screening methods in order to seek or find these sources of resistance within our germplasm. Following that search, we must conduct genetic studies to understand the breeding strategy that has to be undertaken to determine the genetic basis of resistance. And then we establish our pre-breeding cycle or our program, where the interesting resistant varieties that we have located within our accessions in the gene bank are crossed, and these are crossed to develop enhanced population for resistance. And then this takes us to, this enhanced population will take us to resistance progeny with accumulated quantitative genes for resistance, which will therefore mean they have high levels of resistance. And at this step, we hand over to our commercial breeding partners. So up to our pre-breeding cycle is what happens at our gene bank. And these varieties are then used in the support of commercial breeding programs locally and internationally to develop adapted varieties for resistance. These varieties can then be introduced into clonal gardens or seed gardens and used to produce materials for farmers. This is after they have then been properly gone through commercial breeding cycles. Then you get the clones or the seed gardens, and then these can then be released to farmers. The farmer is therefore 6 steps away from resistant varieties that sit in this gene bank. So careful planning is required to keep this pipeline flowing, following and creating new varieties for our farmers. So let's see how this pipeline flows for witch's broom disease. As we know, witch's broom was first observed or reported in Amazonas in the 1700s, and it has been rapidly spreading across South America and the Caribbean, devastating cocoa industries in its path. For example, if we look at the tale of 2 countries, Suriname, which first reported witch's broom in 1895, their cocoa industry was completely devastated, and they did not recover significantly. In 1928, witch's broom disease was first reported in Trinidad and Tobago, and this resulted in a 50% decline in yields by the early 1920s. However, our cocoa industry would have been completely decimated if it weren't for this individual and Dr. Pound, F.J. Pound. In fact, the story of the International Cocoa Gene Bank Trinidad really begins with the search for resistance to witch's broom disease. Dr. Pound, in his search, led expeditions into the Amazon for cocoa germplasm, which showed some resistance to witch's broom disease, and brought them back to Trinidad. These accessions laid the cornerstone for the establishment of the International Cocoa Gene Bank. So our steps in our search for witch's broom resistance our search started in 1998 with support from the American Cocoa Research Institute, which morphed into the World Cocoa Foundation. And this resulted in the establishment of 2 optimized screening methods for witch's broom disease. The first method, the modified spray method, was able to identify genotypes resistant to infection establishment, and it measured infection based on we use this measure as a percentage infection. And the second method identified genotypes which could be rated based on symptom severity, and it is a more quantitative method, able to detect even attenuated symptoms. And together, the 2 methods made the search for resistance more effective at the gene bank. So some of the resistance that we identified from our results with using phase 1 and phase 2 screening, we were able to show that the sources of resistance identified was not, phase 1 screening was not correlated to those identified with second phase screening, which indicated that the mechanisms of resistance that the two types of screening identified were independent and complementary. This slide just illustrates our how we carry out our two-tiered system, screening system for witch's broom disease, where the plants are initially propagated. They are screened with phase 1, which is using a spray screening, evaluated by, for percentage symptoms. Promising clones selected and then put through the pipeline again, where they are repropagated, screened using the agar droplet method, and symptom severity is measured. And this shows the results from the two sets of types of phases of two-tier screening, where we are able to, in the first slide shows you, the first half of the slide shows you infection success in various clones, which shows the greater resistance having lower percentage infection rates, whereas in phase 2 screening, we have the ability to produce attenuated symptoms, even in infected clones that are deemed resistant. So we are able to measure longer infection incubation period and or smaller brooms. And what is the genetic basis of resistance? This we were able to garner from our phase 2 screening. And M by N analysis for genetic resistance showed a predominance of additive genetic effects. And the normal distribution that we saw among the progeny from parental crosses which were done showed that resistance is polygenic. And with this strong parent offspring regression showing progeny performance can therefore be predicted based on parental resistance. And this augurs well for our breeding, since phenotypic selection in early generation, it indicates that phenotypic selection in the early generation will be effective for predicting how our trees behave later on in the field. And so our pre-breeding program was established from 2008 to 2012, and through these crossing of accessions with the highest levels of resistance were done, and we developed biparental progenies that were screened using the two-phase system and as before. And we found that 5% of the most resistant progeny that were planted out in the field, and these trees are now 8 years old, and we are currently evaluating for field resistance. So we go on to black pod disease in our pipeline. How does that flow? And this study for black pod resistance started also in the early 1990s. So we have been able to develop screening methods for determining leaf resistance, pod resistance, and stem canker resistance. Our methods have been able to determine two mechanisms of resistance have been identified in leaf and pods. The first one affects penetration resistance, which is the ability of the pathogen to establish infections in leaf and pod material. And we have shown that this resistance is influenced by issues like stomatal size, stomatal density, and cuticle thickness. And the second level of resistance identified for black pod disease is post-penetration resistance. And this is a resistance that results in pinpoint lesions or expanding lesions, and this is found to have been influenced biochemically. In addition, we have optimized methodology for testing of leaf and pod tissues, and this slide just gives you a sort of idea of how we go about doing the screening on leaves and pods. We have, this is an example of, again, how the tests are carried out. We have our researchers showing how the leaf inoculation and pod inoculations have been done. And this is an example which shows leaf inoculation after punching holes in the leaves. And you can see that in some varieties, lesions do not expand while others do. And in some varieties, the lesions do. So what are the mechanisms of resistance? We were able to garner this from the post-penetration resistance work for black pod disease. And most interesting here is that there is a correlation between post-penetration resistance in leaves and pods. This means that we can therefore predict the lesion expansion rate in pods by punch inoculating leaves. So that meaning that we can inoculate the leaves and correlate how the lesions will expand on the pods of the relevant variety. Just as in the case of which is BROOM disease, we established two mating designs, a diallel mating design and an M by N mating design to do the genetics of resistance. And both showed that post-penetration resistance in leaves had a fairly high narrow sense heritability, which indicates that phenotypic selection would be very effective. And in another study, we also showed that stem canker resistance, it's not presented here, is also determined by additive genetic effects with a narrow sense heritability. So for population enhancement for black pod resistance, we established 99 crosses between the most resistant parents and the progenies. From these were evaluated for post-penetration resistance. The most resistant progeny was selected and planted in the field. And after 8 years, we evaluated the progeny for resistance to black pod by spray inoculation, and the most resistant progeny were then crossed again. Then progeny from these were again screened, and the most resistant plants identified were planted again in the field. These plants that have been planted in the field have also been evaluated to identify the most resistant genotypes. Some of the best progenies coming from 2 recombinant cycles were provided to the Ministry of Agriculture, Land and Fisheries to be used in crosses with the best Trinidad Select Hybrid TSH varieties, that is. These crosses were screened by the Cook Research Center for post-penetration leaf resistance. And the resistant progeny from these crosses were planted in the field and evaluated for pod resistance by the Cook Research Center. The progeny from these show very high levels of resistance to black pod and are also now being evaluated for other agronomic traits in the field. So some of our successes have been due to our networking and collaborating and hard work of many people. Gene banks, like the International Cocoa Gene Banks, are therefore important sources of genetic diversity of cultivated plants and their related wild relatives, as we know. However, in order to exploit this diversity, systematic long-term strategies have to be developed and executed using multidisciplinary teams. Our quest for disease resistance in cocoa that has been carried out systematically over 3 decades at the Cook Research Center is an example of such a multidisciplinary strategy. However, no single person or organization can be expected to do everything, and I would like to thus acknowledge all the researchers who have contributed to our disease resistance work over the years at the Cook Research Center. I have tried to include as many as I can. I am sure I may have left out some names for these. If I have left out anybody, I apologize, but we have acknowledged and appreciate that teamwork that has been happening over the years. Additionally, our success is not only ours, but based on collaboration with breeding institutions, it's also key to make the necessary impact needed. Over the years, the Cook Research Center, Center's pathology team, and as all our teams at the Cook Research Center, has had close working relationships with many other colleagues from other institutes and organizations. And I wish here to acknowledge, especially, and highlight those researchers from the Ministry of Agriculture, Land and Fisheries of the government of Trinidad and Tobago, who have worked over the years on the breeding program to develop blackpod resistant varieties. Again, it has been a teamwork, and we wish to acknowledge everyone who has worked with us to make this happen. I want to thank you for your attention and for joining us in our first Cocoa Research online cards seminar series. Your interest and support of the Cook Research Center is, as always, extremely appreciated. Thank you very much.